Hi, welcome to this Year 1 Essentials video. This is a chance to think a little bit about bacterial cell structure and some important antibiotics targets, and is one of several videos aimed at helping you prepare for the end of year test. So what we're going to do is we're going to go through and do a little bit of revision and a little bit of a draw along tutorial. And this is one of a few videos, and I recommend having a look at some of those. And this is all linking back to the little checklist which I've produced, which is hopefully a way of allowing you to kind of prepare for the exam and for the end of the first year. So in this video, we're going to focus on the top few points. We're going to draw a bacteria. We're going to think about the internal structure and what makes a bacterial cell grow and survive, and think about one or two important pathogenesis factors. And then we're going to label on how certain antibiotics or certain groups of antibiotics work. And this will all be accompanied by separate antibiotics videos and also other videos, for example, on the cell wall. At this point, what I would recommend you do is pause the video and just spend 5 or 10 minutes just sketching out what you already know. Okay, so draw a bacteria, think about what's on the outside of it, the inside of it, and then try to see if you can already remember how these antibiotics work. And then press play and we'll go through it together. Okay, so we'll start by just drawing ourselves a fairly generic bacterial type shape. And we'll start by working from the outside and going in. We'll start with the cell wall. The cell wall is a really important structure within bacteria, and it allows us to differentiate them into two of the most important groups, which are the gram-negatives and the gram-positives, and there'll be a separate video on this. I'm going to put a little star here, and the star's meant to represent the fact that not all bacteria have a cell wall, and those that and there are a small number that do have a cell wall that's neither gram-negative or gram-positive. Bacteria have a membrane, and I will show this as a little dotted line just to differentiate it. And the cell membrane is the lipid bilayer that surrounds the cell. And again, for simplicity, I'm not going to show it, but there are going to be various transporters here, some which kind of span this gap, and this allows solutes in and out. It's also the site of the respiratory chain. So essentially what this is the complex of proteins which allow bacteria to breathe. And remember that all bacteria, well, respire in different ways. Some can grow with oxygen, some without oxygen. That very much depends on the cell type that we're talking about. What else do we have? On the outside of the cell, we're also going to have, or you may have drawn, some specific molecules on the outside. So you may have drawn LPS, for example. This is lipopolysaccharide. And remember that that's only found in the gram-negatives. You may have also potentially drawn LTA. That's only found in the gram-positives. These are really important integral parts of the cell wall which keep the cell wall intact, but themselves can also facilitate disease. And they go by the name endotoxin. In particular, LPS. LPS is really synonymous because if you inject people, when probably you would never do this experiment, but if in animal models, if LPS is injected into an animal model, you become essentially septic. You see activation of the clotting cascade and a systemic inflammation, which is a sepsis-like reaction. What else are you going to have? On the outside, you'll probably have other structures, maybe a bit bigger. And these are adhesive molecules, adhesive proteins, which are just generally called adhesins. Imagine this is a little bit like Velcro, which allows the bacteria to attach to particular cell types. Again, the word adhesin is a really kind of massive umbrella term for a whole family of different adhesive molecules on the outside. And then the last adhesive structure, which is significantly bigger, are the pili. Sometimes these also go by the name fimbriae. And that's a slightly older term. Pili are enormous protein tubes which allow bacteria to attach to surfaces. They can also move, and they also allow for the dissemination of plasmids, which are circular parasitic bits of DNA inside the cell. Let's go into the cell. So we'll have ribosomes. So bacteria will synthesize ribosomes. Sorry, they'll synthesize proteins using their ribosomes in the same way that, or a very similar way to which your own cells work. And remember that the ribosomes are made up of a large and a small subunit. Actually, when you look into some of the details, the large and the small subunit themselves can be further subdivided, but we don't need to worry so much about that. The large is a 50S subunit, and the small is a 30S subunit. And the significance is that although they have ribosomes, they're different in size to your own. And so the prokaryote ribosome can be targeted with a drug. We'll have a nucleoid. They don't have a nucleus like your own cells, but the chromosome is condensed into one part of the cell. So in a sense, it's the instructions to build a new bacterial cell. And this is the thing where you'll see your transcription will happen, and also when the cell is dividing by binary fission, this is the thing that's duplicated, is then segregated down to the other side of the cell, and then the cell splits in the middle to form two new cells. What else are we missing? Well, if we've got endotoxin, there's one other thing that we may have considered, and that is we may have other toxins or other factors present in the cell, and these are sometimes called exotoxins. This is something we'll come on to more in year 2, but you should just be aware of the fact that bacteria can pump proteins out. They can kind of deliver molecules into their external environment. And that's part of the reason with how certain pathogens actually facilitate disease. They actually manipulate the environment around them for their own benefit. And they can do that by using these so-called exotoxins. You may have been thinking nucleoid. I've already kind of mentioned it a little bit up here, but we also have plasmids. So these are smaller circular bits of DNA. And these can hop through pili and then be delivered into other cells. And that allows for genes to be swapped between cells. So for example, antibiotic resistance genes. What else might you have drawn? You might have drawn a flagella. Here, I've just drawn a little box to represent the motor. And again, I suppose the point here is that certain bacteria are motile, and they can swim toward and away from certain attractants and toxins. So they will move away or towards things in depending on their environment. But they don't always have flagella, so not all bacteria can do these things. And then the last thing is you may have drawn on the outside a capsule. And this is a layer of exopolysaccharide or slime, and this helps protect the cell. It helps protect it from environmental danger and stresses, but also it helps hide the cell and make phagocytosis a bit more difficult. Bacteria that can make a capsule are called encapsulated. And the capsule is a really important target in terms of certain vaccines. And there are certain antibodies which will bind to the capsule, and you can be vaccinated against different types of capsule molecules. So when you're vaccinated against organisms like Streptococcus pneumoniae or the pneumococcus, you're actually vaccinated using different capsule molecules. So there we have it. There we have a basic bacterial structure. A couple of important things to say, things which hopefully you've not included, which I'll put down here. No mitochondria. Remember that mitochondria are in effect bacterial cells capable of, well, they have their own ribosomes, they have their own little genomes, and sometimes some of the toxicities of certain antibiotics happen because the drug that you prescribe to treat a bacterial cell cross-reacts with the mitochondria. And also, there's no mesosomes. What's a mesosome? A mesosome is an infolding of the inner membrane, which is kind of often drawn a little bit like this. And it was spotted in, well, a few decades ago when people started to use electron microscopes to look at the inside of bacterial cells, and they saw these structures appear. What we now know, or what we now suspect, is that these were actually artifacts from the microscope process that actually involved quite a lot of dehydration of the cells before they went under the microscope. So they don't really represent anything physiologically useful or relevant, so we can get rid of it. And it's a bit of a mystery, to be honest, about why they're still mentioned in A-level textbooks. What about the antibiotics? So for the last bit, where do the different drugs work? Beta-lactams, such as penicillin, work at the site of the cell wall. So these are drugs which basically work to inhibit the cell repair mechanisms going on. I'll cover this in another video. There are certain antibiotics mentioned which are active on the ribosome. So macrolides work on the large subunit. For me, this is quite an easy one to remember because you just think macro basically means large. We mentioned aminoglycosides. They work on the small subunit. There was also a drug combination which you may not have heard of before called trimethoprim sulfur methoxazole. Sometimes this is abbreviated to TMP SMX. Two very important antibiotics. They can be used individually, but they sometimes are prescribed together. And what they do is they stop the conversion of folate, which you get from your diet, but also bacteria get from their diet, to something called THF, which stands for tetrahydrofolate, because this thing then goes on to form certain amino acids and certain nucleotides. And what this antibiotic does is it stops that from happening, and eventually leads to the starvation of nucleotides, so the cell can't replicate very easily or at all, and certain amino acids, so it struggles to build proteins. So really, this thing works all the way up here. Very important antibiotic or combination drug to work. It does see some activity as a consequence in your own cells, so it does work on eukaryotic cells as well, and you occasionally see this used to treat non-bacterial infections, so it can be used to treat certain fungal or parasitic infections as well. There was metronidazole. This is only effective against anaerobes. And what it does is that it enters the cell as a prodrug, is switched on by interacting with the anaerobic respiratory chain, and then in its active form goes off to damage the DNA. So it actually takes place in two parts. Activation at the respiratory chain, which, because anaerobes respire in different ways, this is how you selectively target these particular bacteria, and then it damages and destroys the DNA. What else was there? There was lastly the quinolones and the fluoroquinolones. And these work by inhibiting one of the enzymes required for DNA repair. So we've actually got quite a few antibiotics selected. There are more, but just as a kind of a starting point, this is a useful place to begin. So we've gone through, from working from the outside, we've considered the cell wall, bits to do with how the bacteria adhere to surfaces, one or two other virulence factors, so one or two things which help the bacteria actually cause disease. So thinking about some of the endotoxins and exotoxins, one or two other features like the capsule, which helps protect the bacterial cell, and then one or two internal structures and things that we've seen as well. Naturally, if you have any questions, don't hesitate to get in contact.